We identify the genes that are responsible for a disease, and we are able to identify genes that other methods cannot see. And that enables us to look at the proteins that these genes code for, and then we could find drug targets among those.
If you had to talk to your grandmom or you had to talk to a kid and you want to explain what biotech's AI does today, it works in the field of genomics, but what it is that it does in a nutshell.
All right, so I just mentioned the Human Genome Project, and if the Human Genome Project had been 100% successful, then now we would have been able to cure all the diseases in the world. But this, of course, hasn't happened. There are some diseases that we were able to cure through it, but others not. And the reason for that is that genetic and genomic data is simply incredibly complex to analyze because it is not big data, but it is wide data. The difference is simply in big data, you're learning something really simple and you have millions of examples to learn it. Whereas with wide data, you're learning something incredibly complex and you only have very few examples to learn from. So the thing here, a good example for big data would be Twitter data. You have billions of tweets. Each tweet only has about 20 words. Whereas in genomic data, the human genome consists of over 4 million genetic variants that are captured in the modern biochips. But then the number of patients you have in each study is only a few thousands, and in cases of rare diseases, only a few hundred. That's where biotech's AI comes into play. We are able to make sense of such data and have, developed workarounds to tackle that data.
Can you give us a sense of what it means with a tangible example? So what is the input and the output of a project where biotech's AI makes a difference?
The general premise of biostatistics, comparing the DNA of a sick cohort and a healthy cohort, and then use that to find the differences that determine whether somebody is sick or not. With the standard approaches, you have huge trouble. You only identify certain single gene variants, but in reality, really, many diseases are polygenic in nature, meaning that there are complex genetic patterns that cause the disease. And our machine learning algorithms are able to identify these more complex patterns. So then that gives us, first of all, a way to predict who's going to get the disease and who won't get it very accurately. But second, also, imagine we have these different gene variants that cause a disease or are associated with the disease or correlated with it. Then you can also look at the proteins, because every a gene variant lies on a gene, and a gene in turn codes for a protein. So then you can look at the whole problem in the protein level, and then you see the metabolic pathways that affect the disease, and then you also see potentials for drug targets, because in those pathways there will be receptors, there will be drug targets, and then you can make your way towards a drug target. We identify the genes that are responsible for a disease, and we are able to identify genes that other methods cannot see, and that enables us to look at the proteins that these genes code for, and then we could find drug targets among those, and we have. Then yes, you could say if there is not a molecule that exists, then you would have to go to a company, for example, that works in designing drug candidates. But the nice thing is also that there are already a myriad of compounds existing in this world, and we have a database on all that. So what we're trying to do at first really is to see, is there an existing drug or existing drug candidate that was maybe used for another indication, or was never released because it didn't work for the original indication, and we look if something like that already exists, and that makes the whole process a lot easier, because then we suddenly have a drug that already exists. We have established that this drug, well, we already know that that drug affects the drug target, and what we have established is that the drug target is actually connected to the new disease. So you could skip all the preclinical testing, and you could simplified, saying this simplified now, go right into a clinical phase 2B. So that is drug repurposing, which is very, very attractive to companies like us who are smaller.